Today I'm going to talk to you about olfactory perception, how smells are perceived and utilized in our brains. So, how do we smell? Pleasant, hopefully. Every day we wake up and experience the world with our noses, along with all of our other senses. We experience things like morning breath, toothpaste, or my personal favorite, coffee. Perhaps, if you're very lucky, you get to smell fresh bacon in the morning. In fact, maybe what gets you out of bed is knowing you get to open up your favorite box of cereal. Imagine if, for that instant of time, that you had no other senses, only your sense of smell. As soon as you opened that box and you got the first whiff, many things would start to happen. First of all, you would know that was your favorite cereal. You would also know just about how far away you were from it. You might start to get a picture in your mind's eye of a time that was tied to that scent. And you'd probably start to salivate, a direct physiological response, as your body prepared for much anticipated digestion. So how is it that all of these things happen exactly? First, volatile hydrophobic molecules leave the surface of your cereal and enter your nose when you breathe in. These molecules then dissolve in a mucus layer that is created by Bowman's glands in order to interact with olfactory neurons. Olfactory neurons are special because their dendrites can interact with molecules outside of the epithelium. Odor information is then propagated up through your cibriform plate that is located at the top of your nasal cavity and is about level with your eyebrows. Olfactory axons carry this information to olfactory bulbs, where it then synapses on mitral cells. The stimulus travels from glomeruli to the olfactory tract, where at the anterior perforated substance it splits into two parts. The medial stria carries the information across the anterior commissure to the other side, and the lateral stria takes that information and brings it up to the primary olfactory cortex. From the primary olfactory cortex, information is then sent to many other different parts of the brain. Parts like orbitofrontal cortex, thalamus, hypothalamus, and the hippocampal formation, which I'm sure you're familiar with, as these areas are involved in memory and appreciation of olfactory stimulation. Okay, so that was easy. What is so special about smelling anyway? Well, actually, olfaction is quite complex, and there's many things we still don't understand. But what we do understand is pretty cool. Like, your sense of smell is the first sense to develop after birth, and it's hardwired into your limbic system, which explains why smells can elicit such strong emotional responses, or flight or fight stimulation. We have a small number of receptors, but we can perceive thousands more scents. We only have about 500 to 750 olfactory receptors, of which 75% do not encode full proteins, so really it's more like 100 to 200 receptors. But an adult human can perceive about 10,000 different scents. Our olfactory epithelia helps us enjoy what we eat. Without that, we would be limited, limited to the 5 tastes that our taste buds perceive: salty, sweet, sour, bitter, and umami. Your olfactory nerve can regenerate, and often does in fact. It is the only neuron that gets replaced regularly in your body about every 4 to 8 weeks. Perception can change with concentration, meaning different receptors are activated at different concentrations, which explains why your perception of a smell can change as it gets stronger or weaker. Pheromones, quite a hot topic right now. Many laboratories are currently working on it. Our sense of smell helps us to identify foods, mates, danger, and the spaces around us, and it is strong. And you might be tempted to argue with me on that last point. How do you think that our sense of smell stacks up to a tracking dog, for instance? Not too shabby. On the left is a picture of a tracking dog tracking a pheasant that had been dragged through a field. On the right is a human tracking the scent of essential oil that was dragged through a field. Yellow lines indicate the actual path of the scent as it was taken through the field, and the red lines indicate the path of the dog or the human as they track that scent. Now, it is true that the surface area of the epithelia does matter, and in dogs it is much larger than in humans. But studies that blocked out hearing and sight showed that humans can track a scent, and that their ability gets better with practice, a consequence of neuroplasticity. These findings suggest that the poor reputation of olfaction may, at least in part, be a reflection of behavioral demands rather than ultimate abilities. In other words, we don't have to track chocolate through a field, but if we did have to, we could. As I said earlier, olfaction is complex, and at the molecular level, it's no different. Aromatic molecules activate subsets of amino acid residues within binding pockets of different odor receptors. Now, which different amino acid residue is activated and in which binding pocket depends on individual properties of the molecules themselves, such as size, functional groups, or the length of their carbon chains. Take the molecule that is present in an orange, for instance. It is the mirror image of that which is present in a lemon. That's how specific it gets. As we all know, no one person is exactly the same as another. There are many variations in olfaction from person to person. Anosmia is defined as the absence of the sense of smell temporarily, permanently, progressively, or congenitally. We've all experienced temporary anosmia, inflammation or infection of your nasal cavity, your favorite seasonal cold. Permanent is caused by head injury or tumors. Progressive is caused by neurodegenerative conditions such as Parkinson's disease or Alzheimer's, in which the patient slowly loses their sense of smell, but rarely notices it. Congenital is caused by genetic conditions, such as Kalman syndrome, which is the failure to start or finish puberty, and primary ciliary dyskinesia, which is the defect in cilia causing it to be immobile. Additionally, opinions of a scent will vary from person to person. What smells nice to me might not smell so nice to you. There are over 100 different point anosmias. Alicent anosmia is the inability to smell garlic. Eugenal anosmia is the inability to smell cloves. Olfaction is a complex and very compelling area of study that began with the likes of Aristotle as far back as 350 BC and is continuing strong today. So keep your nostrils open for what might be discovered next.